Hello, my name is Roxana Bujak, and I'm going to talk about volumetric visualization of the genome. The genome is the complete set of DNA in an organism. It contains instructions for building and maintaining that organism. It is a chain of about 2 nanometers thick, 2 meters long, folded into a sphere about 6 micrometers wide. The specific shape of this folding decides which genes are accessible and can be turned on or off. And that is important for cell identity because different folding patterns result in different cells, even with the same DNA. For example, a cell becoming a cell of a muscle or a cell of a neuron, that is only based on the folding. It is also important for disease because misfolding is linked to cancer, developmental disorders, and aging. But visualizing this is actually not so easy because it is kind of equivalent to folding a 100-kilometer long hair into the size of a basketball. And human perception, on the other hand, is perfect to distinguish and recognize objects that are simple, regular, or symmetric. As an example, most of you probably recognize the butterfly. Some of you even might have said, "Ooh, it's a blue morpho." Whereas the thing on the left, I suppose many of you would have thought, hey, is this maybe the biggest ball of twine from Cocker City, Kansas? And totally not. Clearly, it is the biggest ball of twine made by a single person in Darwin, Minnesota. See what I'm talking about? This is two things that just don't go together very well. So the idea for this paper was, if it is so hard to draw this hair, why even try? Instead of drawing a one-dimensional curve embedded in 3D space, why can we not just draw the volume that this hair actually occupies and the variables that come with it? This can produce very sparse and intuitive visualizations. It allows interpretation by a human, and it also allows for the application of common visualization filters and metaphors. How can we get a volume from this line? There are pretty much two common ways. One is a Delaunay tessellation, where we then have the option to interpolate linearly inside each of the tetrahedra, which is great for drawing a convex hull, clipping, cutting planes, or isocontours. And the alternative is a Gaussian sampling. We add a Gaussian at each point. This volumetrically fills the bounding box with a density, and this is good for thresholding or for a detailed volumetric extent that also allows the existence of holes and cavities. Our collaborators are subject matter experts in epigenomics, and they are trying to understand the dynamic structure-function relationships within the mammalian genome in response to viral infection. What this means, they have infected vero cells derived from the African green monkey's kidney cells with the modified vaccinia virus. And what they try to understand is, how does the configuration of the genome change under this infection? One application scenario is variable comparison. From the experiments, our application scientists have derived 4 variables that they would like to look at the same time to compare if everything works fine. Those are the peaks, the FPKM, which stands for fragments per kilobase per million map fragments, the eigenvalue, and the compartment. Interpreting the DNA volumetrically allows us to apply many different visual metaphors, and that in turn allows us to encode all those 4 variables at the same time without interfering visually. Let's start at the bottom. The tubes describe the actual path that the DNA takes, and the color of the tube encodes the peak counts, and the radius of the tubes encodes the FPKM. Then the Gaussians, what we did is we thresholded the A compartment from the B compartment and then applied a Gaussian sampling on each of those separately. So these transparent clouds, they tell us now what is A compartment, what is B compartment. Hopefully the A compartment coincides with the high peak counts and coincides with the high FPKM. And then on top of them, we have the Delaunay tessellation, which we used first of all to plot the convex hull. So we get an extent of this DNA ball here. But we also used it to draw an iso contour that is supposed to separate the A from the B compartment. And that encodes actually the zero iso contour of the eigenvalue of the singular value decomposition. So now we can see all of these things at the same time. This application is even more challenging because the scientists really try to understand how the infection affects the folding and the shape of the DNA. So what they really have is a control set where it's only mock infected, so it's infected with an actually like zero virus, and they have the one that's actually affected with the virus, and they track those over time and want to see how this changes the configuration. So what these visualizations show here is on the top, the cyan yellow image shows the configuration of the infected sample after 48 hours, where the yellow part is the active compartment, the cyan part is the inactive compartment, and you can see this white isocontour is the same as before, is the part that separates it. And on the bottom, we have the untreated, mock infected alternative of the same type of cell. Here, the active compartment is red and the inactive compartment is blue, and this grayish surface forms the difference. If you now wanted to show those things on top of each other so that the scientists can actually see the real shift in the boundaries where everything is, then if you visualize the tube itself, this is completely impossible to do the task. We're trying to do it as sparse as possible so that we can then overlay it. So what we show is only the convex hull, where the surface is painted in the color that belongs to the compartment, and the surfaces inside, the two surfaces that show where the separation is between active and inactive compartment. And this now is like a sparse enough representation so that you can actually look at them at the same time. And that already brings me to the end of my talk. In conclusion, the visualization of the genome helps scientists understand why cells with the same DNA have different forms and functions or certain diseases. But the genome is very, very thin, very long, and super, super tangled. And a human's visual system can just not interpret this type of data very well. And so what we're saying is, why even try? Instead of interpreting it as a very long, thin, tangled thing, interpret it as a volume. And this volumetric visualization can consolidate this conflict. It also has the benefit of emphasizing 3D co-located features, which would be lost in a one-dimensional visualization. Further, it allows for the application of common visualization filters and metaphors like clipping, cutting, thresholding, isocontouring, everything. And therefore, it can produce very sparse and intuitive visualizations that allow simultaneous observation of very many variables and even comparisons with overlays. Thank you so much for your attention. And yeah, I'm not there. So yeah, I cannot answer any questions. Well, sorry, good luck. If you have questions, just shoot me an email. Thank you.